Okay, good morning, everybody. We're going to get this next session started. I'll just be very brief. My name is Ann Rudy. I'm from Emory University School of Medicine and the Yerkes Non-Human Primate Research Center. The title of our session is New Viruses and Models with a plural S, in keeping with Jeff's introduction earlier to his talk. And our first speaker is Dr. Kristina DeParis from the University of North Carolina School of Medicine. And she'll be talking to us about the need for age-specific non-human primate models to address clinically relevant questions of HIV infection and prevention. And so the way this session, sorry, I should have said this at the outset, the way this session will work will be that we have 5 talks with time for questions after each talk about the specific content. And then at the end, all of the speakers will be invited back up to the front and we'll have a panel discussion. So welcome, Kristina.
Thank you very much. Thanks for the invitation, and I'm glad Jeff went first, and I will try to harp on some of the points that he brought up as well. So thinking about what to present, so these are really the different age groups that we have. And if we look at the 2008 new infection rates, we saw about 10% in pediatrics. About 34% of the new infections occur in adolescents and 55% in the adult population. What I think is really critical for us to remember that among the 34% of adolescents that get infected, 2/3 are young girls. And with young girls, I really mean 15 to 19-year-olds that often have relatively poor HIV knowledge. They are not empowered, and they don't always have access to reproductive health, which leads to a lot of teenage pregnancies. And these young women show up late for antenatal care, show low ART adherence, which brings back the cycle or feeds into the cycle of mother-to-child transmission of HIV. So really, if you look at the pediatrics and the adolescent population together, it's about half of the new HIV infections. So I think that's really important to focus on these two groups, and they are very different from the adult populations. And what I mean by that is if you look at pathogenesis of HIV, obviously in all of these groups in resource-poor countries where we have most of the infections, they're dealing with co-infections. What you have to consider is if you look at pediatric HIV infections, they occur at a time when the immune system and the central nervous system is developing. So there's major damage to the CNS that can occur, developmental delays. I think the same is true in adolescents. They have a higher chance of substance abuse, may it be alcohol, tobacco, or other drugs, which can be associated with CNS damage as well, because even in adolescence, the brain is still developing, and there are hormonal influences too, whereas in the adult population and more senior population, we are dealing with comorbidities. So I want to touch on the infant and adolescents a little bit with regard to where I think the NHP model could really help in evaluating some of the neurological complications that we observe in infants and in adolescents. And so one of the questions that I always get right away is why do you still care about pediatric HIV infection? We have ARTs, we can prevent mother-to-child transmission. Well, we now know that it's definitely true. We have less in utero HIV transmission than you. The majority of infections now occur by breast milk transmission. But you have to remember, sir, and these are the 2018 data, only 50% of all HIV-exposed infants worldwide actually receive a biological test for HIV by 8 weeks of age. And again, this is a time when the central nervous system and the immune system undergoes tremendous developmental changes. So it is a problem. There are still limited ART coverage. Again, the 2018 data show that globally we are at 62% ART coverage worldwide. And that's the whole population. In infants, it's probably closer to 50%. There is still use of nevirapine as first-line therapy in many African countries, if you have been there, which leads to drug resistance. So I think it's a problem we should continue to pay attention to, especially as we have lots of young women that are getting infected with HIV. So we have worked extensively in the SIV macaque model in infant rhesus macaques. And if you infect neonates, or even if you infect infants within the first 12 weeks of life, this is the viremia that you see if you infect infant macaques with SIV MAC 251. If you go back a slide, that's very similar to the persistent high viremia that you observe in human infants and without ART. And there are several studies that have documented in the past that this infant macaque model can be applied to pathogenesis. It has been instrumental in finding out virus dissemination, the involvement of gut in HIV pathogenesis. It has been used for vaccine and cure studies. And it was, Jeff brought up the early ART studies. I think there was so much hesitation initially about using antiretroviral therapy in infants, and Koen van Rompuy studies in the infant macaque model with tenofovir were instrumental in allowing the application of ART to human infants. So coming to the CNS, and that's really not my own data really, but it's really known that approximately 70% of HIV-infected infants have impaired motor skills, cognitive dysfunction, present with memory deficit or verbal deficits. And that's even infants that are on ART. And that is because they are not receiving ART right away. And so there's early damage. So why is that? It could be the timing of the insult because it is during neurodevelopment. It could be that ARTs are not penetrating into the CNS, or it could be because despite ART, there's ongoing virus application. And all these questions have really great implications for treatment of human infants, when we should initiate ART, and it has implications for cure strategies in infants. So in humans, it has actually been shown that certain drugs cannot penetrate into the CNS. And but because there's combination therapy, virological control can be achieved in humans. We can recapitulate this in Rhesus macaques, and this is a study actually by Rudy, where they could show that, and I'm only showing PMPA here, they had 2 other drugs in the study, that similar to tenofovir in humans, there is reduced penetration. The advantage of the NHP model is that whereas in humans, you often only have CSF as a surrogate to evaluate CNS complications, in the non-human primate model, you can look directly in the brain and in other tissues. And I think that's a critical aspect that is very important. And because sharing came up as one of the points, we should share our resources. So in the past couple of years, we have sent all our infant macaque brains to Mark Burkey at Howard University. And he has done studies in the hippocampus and more recently in the dorsolateral prefrontal cortex. And he could clearly show that SIV-infected infant macaques have reduced neuronal growth. And again, this is in ART-naive animals. And why is that important? Because these 2 areas that he focused on, I don't know if my pointer might not be working, the hippocampus is an important part of the brain that controls memory, and the dorsolateral prefrontal cortex is responsible for executive and logical function. So exactly what we observe in human infants. Thank you, Bethel. So I think these studies would also allow us to look at the HIV reservoir, and let me click one more time. Again, I'm coming back to Ann's studies recently, They actually did a study in Rhesus macaques, and they treated those animals with ART, and then they looked for viral reservoir and actively replicating cells. Again, this is something that is more difficult to accomplish in human infants than it is in the Rhesus macaque model, because you have access to multiple tissues, and certainly you could never isolate a brain from a human infant or a human adult. And so what you see, and what is really important, that even when you look in blood or lymph nodes and you look at infants, the majority of the pro-viral DNA was actually found in naive T cells, which is very difficult. Very different, my apologies, to adult Rhesus macaque, where the reservoir is really in the central memory cells. And if you look in the brain and you look for virally infected cells, it's actually in the glial cells, which brings up the still controversial questions: can macrophages serve as viral reservoir? Again, I think the macaque model will provide an opportunity to address that question. And there was another study by Clayman's group that showed that macrophages in the brain can serve as reservoir. And there was a study that was presented at IAS this year from Garner et al., and they really showed convincing data that human urethral macrophages can serve as viral reservoir. Okay, so I think the NHP model can really be used to study CNS involvement in pediatric HIV infection. The advantages I mentioned before is the detailed tissue analysis. You can identify where the insult occurs and correlate it with function. Of course, it is more comprehensive, more difficult to perform these behavioral tests and cognitive function tests. They are doable, but it's more difficult to do that, and that's probably easier to do in human studies. But I think we should not think as one or the other, but again, as really considering the monkey model as being, or the monkey models, to be complementary to clinical human studies. And while I don't have the time, I think we could similarly use the adolescent non-human primate models to study alcohol and substance abuse and the effect on neurological changes in the brain. So in the second part, I want to briefly touch on vaccines, development and testing, which is what our group spend a lot of work on. And because Louis is here, I will really focus on vaccine strategies that have the goal to elicit anti-HIV envelope antibodies, be it antibodies with neutralizing or Fc-mediated effector function or both. And so to do those studies, as Jeff alluded to, the novel HIV envelope shifts have come into the picture. And these are three different shifts that have been developed, and they were provided to us by Dr. Shaw's group, by Sampar Santra, and Bruce Ruprecht indirectly. And I think what you can see right away is that while these viruses can elicit acute viremia at very high levels, chronic viremia is very variable. You can have animals that have persistent high viremia. You can have animals that will go on to completely control, or you will have some animals that go up and down and have fluctuating viremia. So again, as Jeff also, I want to reiterate his points. Are these viruses reflective of pediatric HIV infection in humans? To do that, long-term pathogenesis studies would have to be conducted in non-human primates. Are they suitable for ART reservoir cure studies? At the moment, I would think not, because they can spontaneously control. So those studies need to be performed. This comes back to the question, which model is best for what question you want to address. If we use, however, these viruses, this was a SHIV CH505 provided by George Short and Catherine Barr. If you look at the infection course is totally similar in infant and adult Rhesus macaques. There's really no difference. And if you look at envelope-specific follicular T helper cells or memory B cells or antibody responses in these animals, the magnitude is absolutely the same. There is no difference. However, if you look at the function of follicular T helper cells or germinal B cells, there's a difference between infant and adult macaques. So there might be functional differences that we need to evaluate if we want to use those viruses to test vaccine efficacy, and that will be very important. To optimize infant antibody responses, you can use adjuvants. And our group, together with Sally Permer, we have conducted a study where we tested multiple different adjuvants in parallel. This study has been published, and we could show that adjuvants other than alum, especially 3M057, a TLR78 adjuvant, and stable emulsion, can really enhance antibody responses, not just by magnitude, but also by function. I'm only showing one example here, that is ADCC. And despite these improved antibody responses, we don't actually see an increase in somatic hypermutation. So again, that's different from infants. You have better antibody responses, doesn't necessarily mean that you have higher somatic hypermutation. So there are really age-specific differences in these models that need to be considered. We could also show in our studies that infants are totally capable of eliciting antibody responses, and these are persistent. So these animals were vaccinated at week 0, 6, and 12 of age, and then at week 14, we had peak responses. I'm showing again plasma antibody titers, avidity, and ADCC. You see the responses diminish over time, but if you give an additional boost at week 32, you can absolutely boost those responses, which is consistent that we were able to induce memory in these infants. So you can start immunization early. And again, if you use an age-relevant adjuvant, you can compare to just like a squalene adjuvant, you get much stronger responses. So there are ways to exploit the infant immune system. And because I think I'm running out of time, what I'm trying to say, coming back to my initial point, that I really think that the epidemics of pediatric HIV infection and HIV infection in young women are linked. The goal should really be maybe to exploit early life vaccination to protect young adolescent women against HIV infection, to stop the cycle and prevent them. And so I think we have an opportunity to use the Rhesus macaque model to start vaccination in infancy and then maybe boost later. And when they have reached sexual maturity, which is much earlier in the non-human primate model, we can test if a vaccine initiated in infancy can actually protect against sexual transmission in adolescence. And so my points for discussions are that I was really running through it a little bit, but I just wanted to give you a few thoughts for consideration, raise a few discussion points. To do those studies, we really need larger group sizes in our studies. If we want to do study if we can prevent sexual transmission, gender needs to be considered. So do we have group sizes where we have enough males and females? Are those studies possible? Again, as Jeff pointed out too, those studies would really have to go on for several years. And we need to select the relevant model depending on pathogenesis, vaccination, or cure, and then use new tools. And if we want to do it, we will need a lot more money to do those studies. But I think the NHP model still has a lot to offer for our understanding of HIV infection and how to prevent it. Thank you very much.
Okay. Are there any burning questions yet?
Do you see any difference in the durability of the antibody response in the neonates versus the adult macaques?
So we haven't been able to do parallel studies. So,
okay. So for how long
we
did
have
you
followed our infants for like close to a year, and so you can boost it and you get good responses, and so I think the responses will be durable, and that actually has been shown in humans too now. Jeannie Fowler has done some retrospective analysis of early pediatric HIV vaccine trials, and could show that those responses were maintained for up to 2 years. And some of those antibody responses were as high as you would observe in an adult. So I think, again, magnitude is only one aspect. I think we really have to look at function, and that will be a different question. Mario.
Is the virus that comes through the breast milk and infects the infant in the presence of maternal antibody, is it different in terms of sequence or glycosylation? And how would that impact in the primate model? How do you know which virus to use to model that?
Yeah, so we really use oral HIV infection normally. So Angela Amadei, many years ago, has done real breast milk transmission. So she infected the dams and then co-housed them with their newborns and looked at breast milk transmission is as frequent as you would see it in humans. So this is a model that is very expensive. And so to do the studies that we are doing, we cannot afford to do them, to be quite frank. In humans, we know that the virus that is transmitted from the mother to the infant, obviously the infant gets a virus that has been under evolutionary pressure by the mother. If the virus is transmitted, the maternal antibodies are obviously not protective, only some are. There are studies that show that also maybe the paternal MHC may have some impact. When I say the paternal MHC, because the infant has MHC alleles from both the mom and the dad, and so depending what MHC status the infant has, also the T cell response in the infant will be very different, and that it might be able to mount potent immune responses to the infecting virus or not. But there have been some recent studies that show that the infant is actually able to mount new responses, and those might be to some subdominant epitopes. So I think we are still learning a lot about what is different in the infants and how to modulate those responses and potentially enhance them.